Okay, good morning. Let's get it going. Well, it's not many people, right? You guys really should get extra points. Okay. um, so I think we'll have about 4 lectures left, including today's. And I think you guys have been doing great. You guys are working very hard. So I just wanted to remind you guys to check, your grades and to make sure that there's no mistakes or errors or missing points or something like this, right? Make sure. Okay, so today we're going to talk about the fuel utilization. I just wanted to go over again, at this point, we wanted to divide it more precisely the timing in terms of a fasting condition. So fat condition usually we call called absorptive phase, means that the 2 to 3 hours after meal, during this time period, we usually consider fat condition, right? And for example, you ate breakfast at 8 o'clock and about till probably 11 or so, and then you probably started feeling hungry, right? You wanted to eat lunch. So, but during, between 8 and 11 or 12, this time period, you usually consider fat condition. And beyond that, if we don't eat lunch and, and beyond like 4 hours after breakfast, and that time period is not considering the fasting condition. And we're going to talk about how macronutrients that we absorb and metabolize during this absorbed phase or fat condition, or during the fast condition if we don't eat lunch or something like this. And we also going to talk about how our body utilize the energy fuels different between the different phase, absorptive phase or post-absorptive phase, and how our body know to switch the fuel utilization from exogenous or dietary sources of energy to endogenous source of energy. And also there is kind of switch between the different type of fuels. For example, in the fat condition, mostly we use carbohydrate, right? Because we have a lot of glucose, absorbed from the diet. So we have enough energy from the diet. So usually our body utilizes more carbohydrate in the fat condition, and during when switch to the fasting condition, we utilize more fatty acids or fat as the energy fuels. And we also wanted to, what kind of hormones and transporters and substrate that involve in the regulation of this kind of switch, right? From exogenous fuel to endogenous fuel, from using carbohydrate to using fat as energy fuel. Okay, so a standard meal we eat usually contains the 9 gram carbohydrate, 30 gram protein, and 20 gram fat, just roughly. And so now we talk about absorptive phase, right? So how carbohydrate that we absorb and metabolize, distribute in the different tissues. So first of all, we know already, right? Absorbed it, digested and absorbed by the enterocytes. Enterocyte is very important to absorb the nutrients. And we also know the different transporters, right, involved in the absorption process. And once we eat a meal, immediately, or the most important thing is that we increase the blood glucose levels, right, which will stimulate insulin secretion from pancreas. So how carbohydrate being distributed or metabolized in the different tissues in our body? So first of all, the liver takes up about 20% of the glucose that absorbed it into the body. And we know the transporters in the liver is the GLUT2, right? GLUT2 basically is the glucose responsive transporter. It means when we have a high levels of glucose in the circulation, and this transporter GLUT2 being activated and transport the glucose from circulation into the liver. And glucose, once it enters the liver, glucose can be rapidly metabolized to the glucose 6-phosphate, and the enzyme, that glucose kinase, which is highly abundant in the liver, right? So they catalyze this reaction. So glucokinase in the liver does this function to convert it or to metabolize glucose to glucose 6-phosphate. And most glucose that's taken up by the liver through the GLUT2 is actually used to synthesize glycogen, right, and store in the liver during this time period. And the liver, even in the fat condition, usually although they also complete oxidize glucose to produce CO2 water through the whole process, remember, right, TC cycles, but majority of, or most of the glucose partially oxidized in the liver. And the purpose of this is try to, again, to save the carbon skeleton and to partially oxidize to lactate. And lactate can be recycled, right, to make a glucose through the gluconeogenesis pathways in the liver. Although it's still fat condition, but gluconeogenesis also occurred, right, in the liver. But this gluconeogenesis pathways, the converted lactate to glucose 6-phosphate and to the glycogen, not like a released glucose, right, in the fat condition. So, so again, the main purpose of the major pathways of glucose metabolism once it's entered the liver is to synthesize glycogen, right? So glycogen synthesis pathways being very active in the fat condition, and to make a glucose, to convert glucose to glycogen and store. Here I just wanted to tell you a little bit more about the glucose that be used to synthesize glycogen, and where this glucose come from, basically. So 60 to 70% of glucose that being used for making glycogen actually is directly from diet, right? So dietary glucose. And the another 30 to 40% of glucose for making or used for making glycogen is indirectly from lactate and amino acids, right? Lactate, we know, already explained, liver partial oxidized glucose to make lactate, right? So this lactate being used to make a glucose and store as the glycogen through the gluconeogenic pathway. But amino acids, our diet also contains amino acids, right? So we also, digest protein, absorb amino acids. So lots of amino acids will also be used as the precursors of a glucose, right? And to, again, through the gluconeogenic pathway to make a glycogen. So this sort of the whole diagrams give us better pictures. The dietary glucose, once absorbed it, and actually 20% of absorbed glucose, right, goes to liver. And this 60 60-70% means that the glucose that contributed to glycogen, 60 or 70% is from dietary glucose, right? And this is a sort of pathway, for making glucose 6-phosphate, converted glucose 1-phosphate, and glycogen. And also part of glucose being partially oxidized to generate lactate, which can serve as the precursors for gluconeogenesis pathway. Lactate converted pyruvate and enter gluconeogenesis pathway and produce again also glucose 6-phosphate. And this glucose 6-phosphate will not converted to glucose and release because in the fat condition, the glucose 6-phosphatase activity is very low, right? So that's the reason why the majority of the glucose 6-phosphate will go this pathway for glycogen synthesis, right, in the fat condition, because the glucose 6-phosphatase activity is low in the fat condition. So this pathway will not be occurred in the fat condition, right? Does that make sense? Okay, and in addition to lactate and some glucogenic amino acids that we also obtain from the diet can also serve as the precursors. For example, some amino acids can be converted alpha-ketoglutarate, oxaloacetate, and also enter these gluconeogenic pathways and for the production of glucose 6-phosphate, glucose 1-phosphate, and glycogen, right? So this is the whole pathways and how glycogen is being made in the fat condition in the liver. And not only, again, the glucose, not only from the dietary glucose, but also from the amino acids, right? And 20% of absorbed glucose from the meal is also utilized. Well, actually, we already talked about this, right? 22% of glucose is metabolized by the liver. And brain is going to take up 22%, and about 22% of absorbed glucose for direct oxidation, right? Brain oxidized glucose completely. And other glycolic tissues takes up 2 to 3% of absorbed glucose for metabolism. And most of the glucose is also taken up by the muscle and adipose tissue in the insulin-dependent manner. Remember, in the fat condition, insulin level is high, and the two major tissues that have the GLUT4, which is insulin-dependent, right? Skeletal muscle and adipose tissue. So these two tissues take up glucose in response to insulin, high levels of insulin, right? Take a lot of glucose into the tissue, so then can reduce the circulating glucose level. So that means a lot of glucose being taken up by the two tissue and metabolized in these two tissues. And muscle takes up, the glucose being taken up by muscle, 50% of oxidized, provide energy ATPs, and another 50% actually glucose being taken up into the muscle and will convert it to glycogen and store in the muscle, right? 50%, 50%. 50% utilized, complete oxidized for energy, and another 50% of glucose being stored as a glycogen in the skeletal muscle, right? And adipose tissue takes up glucose, usually also partially oxidized and to lactate. And about the 2% of uptaken glucose in adipose tissue were used for synthesis of glycerol 3-phosphate. Okay? And this we know is the substrate, right, for the synthesis of triglyceride. So in the adipose tissue, in fat condition, some of glucose being partially oxidized to produce lactate, and the other portion of the glucose being used to make glycerol 3-phosphate for the synthesis of triglyceride in the fat condition, right? In the fat condition, adipose tissue also need to store triglyceride, so need substrate glycerol 3-phosphate. And this is, again, made from glycolytic pathway, right, from the glucose. Okay, so here is this diagram to show you how dietary glucose being distributed into different tissue and metabolized in different tissues. In the absorptive phase, mean just 2 to 3 hours, after we eat, and all this glucose being immediately, distributed into the different tissues. So see, the 15 to 20 grams of the glucose go to the brain for energy use, and 20 to 45 grams of glucose being distributed to the to the muscle, and half them for complete oxidation for ATP production, and another half glucose being used for synthesized glycogen and stored in the muscle, right? And 20 grams of glucose go to the liver for glycogen synthesis, and also go to glycolytic pathway to produce ATP. And again, here we don't have the, but the partial oxidized, right? I mean, in the liver, most of the glucose and produce lactate and be converted back to the glucose and stored as the glycogen. And 2 grams of glucose go to the adipose tissue. Some of them for partial oxidation produce ATP, and 2% of them will actually metabolize to produce glycerol 3-phosphate, right, for triglyceride synthesis. Okay, I think this diagram probably is a better view of the, the glucose metabolism in the absorptive phase, after meal, the 2 to 3 hours after meal, right? So we can see the dietary carbohydrate being absorbed at 22% of the dietary glucose that being absorbed and go to the liver for metabolism. And we know the majority of the glucose being converted to glycogen and stored as the energy, right, in the liver. And this can be directly from the glucose, right? Glycogen, the glucose can directly be used to synthesize glycogen, and also indirectly, some of the glucose go partial oxidation and produce lactate, and some amino acids also contribute to the glucose for glycogen synthesis. So both amino acids and lactate being converted pyruvate, and or the other intermediates like alpha ketoglutarate, oxaloacetate, right, enter the gluconeogenesis pathway and convert it to glucose 6-phosphate and synthesize glycogen, right? So this is how 22% of absorbed glucose metabolized in the liver, and that non-uptaken by liver, and the glucose that had not been uptaken by the liver or non-metabolized glucose then go to the circulation, right? So that means the rest of 78% of absorbed glucose, right, go to the circulation. And this circulating glucose, and 22% of circulating glucose, and go to brain for oxidation, and 2 to 3% of the circulating glucose go to the other glycolytic pathway. So almost any type of tissues are glycolytic tissues, right? So it means that go to the rest of the tissues. And other important thing is that these 2 tissues, and skeletal muscle and adipose tissue. These two tissues are insulin dependent, uptaking glucose, right? So in the fat condition, again, the insulin levels rise. So the circulating, the rest of percentage, I guess about, calculation, we can calculate probably 60 something, right, percent, probably more. The glucose, circulating glucose actually go to the skeletal muscle and the adipose tissue, right, picked up by these two tissues, and again depend on the insulin levels. Oh, here the little bit more information about the glucose. And in addition to the glycogen synthesis, some of the glucose will also be utilized to synthesize fatty acids, triglyceride, and this sort of lipogenesis pathway, right? It's also active in the fat condition and released as the VLDL. Right? Any questions about this carbohydrate metabolism in the fat condition? Right? Okay, now how about protein? So how we metabolize protein when we eat, absorb the protein. So we have in our standard diet, we have about 30 grams of amino acids in a meal. So amino acids, after transport across the enterocytes and into the circulation, 30%, 33% actually of the amino acids that we absorb it will be used for synthesis of proteins, right? So about 1/3 of amino acids being used for protein synthesis, and the rest of the absorbed amino acids will be metabolized. So 13% of absorbed amino acids will stay in the small intestine, enterocytes. We know enterocytes need the amino acids, right, for the energy. So like glutamate, aspartic were used in the small intestine, enterocytes, for energy, for ATP production, right, for energy use, and then converted to alanine, proline, citrulline, ammonia, and lactate, and this product will be released into the circulation, right, and from the small intestine. And 13% of amino acids, particularly branching amino acid, will be metabolized in the muscle, right? And the rest, or the other 40 to 50% of amino acid will be metabolized in the liver. So almost half of amino acids that we absorbed are metabolized in the liver. So liver is the most important tissue that metabolizes amino acids in the fat condition, right? So let's talk about the protein metabolism in the liver during this absorptive phase. So liver, 60% of amino acids that being taken up by the liver will enter the carbohydrate, will convert it to carbohydrate and store as glycogen. Remember, when we talk about the glucose metabolism, right, the part of the glucose stored in the glycogen actually from amino acids. So here we see that a lot of, like 60% of amino acids that enter the liver actually converted to glucose and stored as the glycogen in the liver. And liver is actually partially oxidized like the other nutrients, glucose, amino acids also partially oxidized. The purpose is the same, just try to save the carbon skeleton, right? for glucose, save the carbon skeleton for glucose, right? And of course, remember the amino acids molecules also have a nitrogen, right? So nitrogen also need to be metabolized, and usually for nitrogen be break down and used for urine, right? The synthesis of urea. Urea usually, be either get rid of the body, or it's a way to get rid of nitrogen, right? It's a urine synthesis pathways, and yeah. So, and the other things that we actually also talk about in the fat conditions, that the major energy fuels for ATP production in the liver is partial oxidation of amino acids, right? So amino acids is important. The energies for the liver, for ATP production in the liver. All right, so here is a diagram again to show you the distribution or metabolism of amino acids in the body during the fat or absorptive phase. So we have about 30 grams of amino acids that we can take from the diet, and 1/3 of amino acids, right? Like 33%, which is about 10 grams of amino acids will be used for protein synthesis, right? We need proteins, and the 12 grams of amino acids will go to the liver for the metabolism, and we know that 60% of amino acids we go to the glycogen synthesis pathway, remember, right? Of course, that's involved in the gluconeogenesis pathway, right? So first of all, converted the glucose 6-phosphate and then being used for the glycogen synthesis that's stored in the form of the glycogen. And some of the amino acids can be oxidized, and the nitrogen in the amino acids molecules being carried, need to be metabolized through the urine synthesis pathways. And 4 grams of the amino acids, particularly branching amino acids, will go to the liver. Go to the muscle and also kidney for further metabolized, right? And 4 grams of amino acids that stay in the small intestine, particularly glutamate, aspartic, right? This type of amino acids will metabolize in the small intestine for ATP production and release this product. Okay, fat. So yes. The percentage, no. No, you don't need to know the percentage or gram, but just kind of idea, for example, majority of, amino acids in the liver will go to the gluconeogenesis, the glycogen synthesis, something like this. So the percentage or gram just give you idea, which tissue metabolize more, amino acids or carbohydrate or something like this, right? Okay, fat. So most of fat in the meal, actually, we know once we absorb it, it should go to the adipose tissue for storage, right, as the energy, as the triglyceride. And we know the 2 lipoproteins that play a very important role, transport dietary fat to the adipose tissue. Remember which 2? Lipoproteins that transport the dietary lipids to the adipose tissue. Chylomicrons and VLDL, right? So, okay. Um, and one thing that I wanted to point out that the fatty acids level actually is relatively low in the absorptive phase because the majority of fatty acids, once we absorb it, they usually will stored in the adipose tissue, right? So the fatty acids level in the circulation will not that be high during the fat condition. And we talk about this many times, right? So lipoprotein lipase is important enzyme to break down triglyceride and to help the fatty acid uptake by adipose tissue, right? And here just give you idea the changes in the circulating levels of glucose, insulin, glucagon in response to a meal, after a meal in the absorptive phase. We definitely expect that the glucose level will be high, right? And insulin levels also will go high after meal. So usually about 15 minutes after meal, you see the glucose levels rapidly increase and reach the peak levels, right, within the 15 to 30 minutes after meal. And insulin have the same pattern, right? We know that glucose stimulates insulin, so when we have a high levels of glucose, and insulin level should be also high. So insulin have the same profile or pattern and as the glucose, right, also increase within the 30 minutes after meal. And then after 3 hours, 3 hours after meal, the blood glucose levels actually decline and return the normal range, or the range before a meal, right? And similar for the insulin levels. And glucagon levels is just opposite, right? And we don't need glucagon to stimulate glucose production. So glucagon levels is actually a significant job, particularly reach the lowest level at the the 1 hour time point, and then after 3 and 4 hours later, then glucagon level turn back to normal, right? And also some of the metabolic fuel levels in the circulation after a meal. You see that the glucose level, we already talked about it, right? Increase and then decline and return to the normal range. Will not say normal range, to the fasted levels, right? And amino acids, you see also increase a little bit, and triglyceride levels also increase a little bit, but not like sharp increase like glucose, right? And lactate levels is also increase. Why lactate levels increase after meal? Yes, oxidize, right? Lots of tissue will oxidize, but some of the tissue, like red blood cells, right, you oxidize glucose and produce a lot of lactate. So lactate levels also increase, also decline. And ketone bodies and glycerol level were very low, right, in the fat condition, right, because we don't need this energy fuels, right. Okay, now let's talk about the sort of post-absorptive phase or fasting conditions. So what do you need to know about this phase? First of all, the duration. So post-absorptive phase refers to the time from the 3 to 4 hours after meal to days of starvation, right? And so what happens during this post-absorptive phase? So usually, for example, at 12 o'clock we feel hungry and we wanted to eat food, right, in lunch. But if we don't eat lunch, what will happen in our body? So that means there is a switch in fuel utilization. There are two aspects in terms of the switch of fuel utilization. One is the switch from exogenous fuels to endogenous fuels. Means that if we don't eat food, don't eat meal, then we need to utilize the endogenous fuels, right? So this is switch. In the fat condition, we usually utilize the dietary nutrients, and in the fasting, then our body need to switch to utilize endogenous fuels. And the second aspect of this switch is the switch from utilizing carbohydrate to utilizing fat as a major energy source, right? So if you guys all know, during the fasting, right, lipolysis is increased, right? HSL activity is increased, so stimulate lipolysis and release a lot of fatty acids from adipose tissue, and lots of tissue will utilize fatty acids as an energy, try to spare glucose, right? So in the fasting condition or post-absorptive phase, then our body uses more fatty acids and save the glucose. And there is sort of a random cycle. It's a theory to explain the fuel utilization switch in the starvation. So we have sort of molecular mechanism to explain how our body switch the energy, utilizing energy fuels, right? And how does the body accomplish this fuel switch? So that means we also need to know the regulatory mechanism, how our body to make this change, right? So basically they regulate by hormone substrate, so we need to know this insulin, glucagon, catecholamines will involve in this regulation. And also substrate, the two important substrates, malonyl-CoA and acetyl-CoA, right, also serve as important regulators of the fuel utilization switch, right? And here's more information about the glucose, insulin, and glucagon levels in the circulation during the prolonged starvation, right? So you know that you see the couple hours to days, like 7, 14, 20, but I don't think it's critical, but just gives idea, how these hormones and substrates and nutrients that change the level in the circulation, right? So see the glucose significant drop, insulin dropped, and glucagon levels significantly increase, right, during the starvation period. And then we're going to go into the more detailed and put all this like absorptive and post-absorptive absorptive phase together and divided this time period into the 5 different phases, right? Including the phase 1 basically is the absorptive phase, right? Like 2 to 3 hours after meal. And phase 2, so start from phase 2, phase 2, 3, 4, and 5 is considered the starvation period, right? And so the early starvation, intermediate starvation, and prolonged starvation period, right? And then we'll see how our body utilizes different energy fuels. And this diagram only show you actually glucose metabolism. So I just wanted to add 2 more things. This is actually very important, right, during the prolonged starvation, the fatty acids and ketone bodies. And so, for example, fatty acids, or probably a little bit earlier, and then body starts to utilize fatty acids. Right? In the prolonged starvation, for example, 48 hours, two days later, then our body, the ketone body, become very important energy fuels, right? And during the starvation. So as you can see here, in the in the absorptive phase, also consider the fat condition, the phase 1. Our body use the energy fuel majority, the primarily actually from the diet, right? So exogenous fuels are the most important fuels in the phase 1 or absorptive phase, right? And the phase 2, that means we start starvation. So phase 2, you see the glycogen is very important energy fuels, right? So here I wanted to remember the time point, when glycogen is completely depleted, right? Like between 16 to 18 hours. After 16 or 18 hours, the glycogen is completely depleted. Right? So the phase 2 basically is 4 hours after meal, from 4 hours after meal to like about 16 hours after meal, right? So during this time period, in phase 2, and the glycogen is the most important energy source, right? So we break down the glycogen and utilize as energy fuels, right? And the third phase, phase 3, after 16 to 18 hours, and because of glycogen is depleted, right? So our body need to make new glucose. So gluconeogenesis become very important sources of glucose, right? So you see here, and I think I probably need to shift this fatty acids line till like phase 2, right? So start from phase 2, actually fatty acids being released from adipose tissue and serve as sort of alternative energy fuels, right? And after 24 hours, and you see the utilization of fatty acids is significant increase, right? So after 24 hours, the fatty acids after 24 hours of starvation, fatty acids become very important energy fuels, right? And then ketone bodies, the time point was like 2 days, right? Remember, 2 days after starvation, and ketone bodies, become the very important energy fuels. All right. Do we need a break? All right. 10 minutes break. Okay. So we already talked about the 5 different phases, and in each phase, what kind of metabolic fuels our body utilize. And I also mentioned the couple time points that you guys probably wanted to remember. One is the 16 to 18 hours. It's a time point that our body glycogen is almost completely depleted, right, during the fasting. And the other time point, like 24 hours, for starvation, that fatty acids become a very important alternative metabolic fuels. And 2 days of starvation, the ketone bodies become a very important energy fuel. And this table tells us particularly the glucose utilization that's been changed during the 5 different phases, and in particularly in the brain, what kind of fuels that in terms of the energy metabolism that brain uses, right? So first of all, the stage 1. Let's see. So stage 1 basically is a fat condition, right? It's like 2 to 3 hours after meal, and the dietary carbohydrates are the major source of energy for all the tissues, right? All the tissues utilizing the glucose as the energy fuels in the first phase, and for the major fuels of the brain, and of course brain utilizes glucose in the first phase, and as phase 2, glycogen, particularly hepatic gluconeogenesis, produces glucose, are the major sources of glucose particularly for the brain, right? And in terms of for the other tissues, the utilization of a glucose, the rate of utilization of glucose actually decreased, right? The purpose of this, we know that just try to spare glucose, right, for the important tissues, brain and red blood cells. And the phase 3, the gluconeogenesis then becomes more important as compared to glycogen in terms of the glucose, right? Because of we use up all the glycogen, right? So we need to make new glucose. So hepatic gluconeogenesis, the gluconeogenesis in the liver has become most important to making new glucose, to contribute to the glucose utilization. Again, and the phase 3, brain still, the glucose still the major fuel of the brain, but the other tissues that start to decrease, right? It's already decreased in the phase 2, but continuous decrease the utilization of glucose in the other tissues, right? And phase 4, the gluconeogenesis is very important, right? Even more important than the phase 3 in terms of the glucose source, right? And here the kidney start also becoming most also important sources of glucose, of gluconeogenesis, right? So, and the phase 4, brain still use the glucose as a major energy fuels, but ketone bodies are also become more important for the brain, right? So both glucose and ketone bodies are equally important for the brain in phase 4, right? And phase 5, glucose still, the gluconeogenesis from kidney and liver still is a major source of the glucose, right? The phase 5. But for the brain, the ketone bodies now become more important energy source compared to glucose, right? And here again, this table just tell us the glucose, right, utilization. I did not mention the fatty acids and ketone bodies, also ketone bodies here. But just keep in mind the fatty acids as very important alternative fuels, right? And this is just describe more detailed, different phase. Phase 1 is absorptive phase. Phase 2, the post-absorptive, and phase 3 is the early starvation. 4 is intermediate starvation, and 5 the prolonged starvation, right? Any question about this table? Right? So again, our body utilizes during the starvation the glycogen, using glycogen first, and then make a glucose, gluconeogenesis, right? And liver plays important role in the early stage of starvation, and later on the kidney will also become more important to the gluconeogenesis. Okay, now let's switch to the metabolic fuel utilization. Metabolic fuel utilization switch, regulation of the fuel utilization switch. So this switch usually occurs in the fasting condition, right? So during the post-absorptive phase, for example, 4 to 18 hours after meal, and this we usually define a phase 2, right? Remember. So blood glucose level is maintained, basically is from the glycogen breakdown, right? Liver glycogen breakdown. So liver glycogen breakdown is the most important, at least glucose is the most important source of glucose. And in this phase, the brain accounts for like 50% of glucose utilization. So in the phase 2, during this time period, 50% of a glucose actually goes to the brain, used by the brain. Right? And the utilization of glucose in the other tissues actually start to slow down and try to spare glucose for brain and other tissues like red blood cells. And after, I think I probably need to collect. And remember the last slide I told you the 16 to 18 hours, right? The glycogen depletion. Here I think probably kind of broad range of the timing. And this slide I show you like 24 hours after starvation. So I think it will still stick on the 16 to 18 hours, all right? I will make correction for this slide later on. So again, after 18 hours starvation, liver glycogen is depleted, right? 18 hours is the time point, right, for the glycogen depletion. Now, what happens after 24 hours starvation? Then we enter sort of a phase 3. First of all, the gluconeogenesis is the only source of glucose because of glycogen has been depleted, right? We don't have a glycogen store anymore. after 24 hours. So gluconeogenesis becomes most important and becomes only source of the glucose. And the precursors for gluconeogenesis can include a lot of stuff, right? A lot of substrates like glycerol, pyruvate lactate, amino acids will all can be the precursors of the gluconeogenesis. And here's a question. Is the acetyl-CoA able to make glucose? Can we use the acetyl-CoA to make glucose? Or acetyl-CoA is glucogenic? No. Yeah, no, thank you. So it's acetyl-CoA only have 2 carbons. I think I mentioned usually 3 carbon molecules are able to use for make glucose, and acetyl-CoA only 2 carbons, right? So acetyl-CoA is not able to make glucose, right? We cannot use acetyl-CoA to make glucose. And during this time period, the other glycolic tissues will continue to use glucose, but the rate of utilization actually decrease, right? And the purpose is, again, just try to spare glucose. A lot of and also a lot of glucose. The glucose is metabolized in a lot of tissues. Also, just goes only partial oxidation to save carbon skeleton for making glucose, right? So glucose partial oxidized in a lot of tissues at this stage for making lactate, and lactate can be served as the precursor for gluconeogenesis, right? So brain is the only tissue that still complete oxidized glucose, right? At this phase 3, after 24 hours, brain still utilize glucose and still able to complete oxidize glucose. Okay. So I guess that's pretty much all for this slide I wanted to point out. One question about where do the amino acids for gluconeogenesis come from? We know that amino acids is one of the precursors, right, for gluconeogenesis. Where do these amino acids come from during the starvation? Muscle breakdown, right? So from the muscle. And this slide just continues. So during this time period of phase 3, or after 24 hours starvation, the fatty acids now become the major fuels for many tissues, right? Like skeletal muscle. And ketone bodies start to be synthesized and used as a fuel for brain and skeletal muscle. And kidney also now becomes important sources of glucose, right? Kidney can also make those gluconeogenesis and make glucose. All right, so here again the concentrations of the energy fuels during the prolonged starvation. You can see the ketone bodies now significantly increase, right, in the circulation during prolonged starvation and become the most important energy fuels. And I listed here some of the energy fuels decrease and the other alternative energy fuels, they increase. For example, glucose, amino acids, lactate levels are decreased, right? Because we try to use less glucose, right? So lactate levels decrease, and you also try to use less amino acids. And then the ketone bodies, fatty acid, triglyceride levels are increased. These are the most important alternative fuels during starvation. So we will expect the ketone bodies, fatty acids level, triglyceride level will be increased during the prolonged starvation. Okay, now let's talk about the Randle cycle. So this is a theory to explain how our body switches energy fuel utilization, right, from fast state to the fasting conditions during the fasting. So this is the Randle cycle is a metabolic process that involves the sort of competition of the glucose and fatty acids for utilization in the starvation. Also in some kind of a disease condition like type 2 diabetes, insulin resistant, but we can just focusing on the physiological conditions, starvation first, right? So that means and this theory actually being described in like 1963, by this scientist, Philip Randle. So that means in the fat condition, our body mostly utilizes exogenous fuels and primarily carbohydrate as the major energy fuels. And during the starvation, our body utilizes more fatty acids and save glucose, right, in the starvation. So this kind of switch, is actually there is sort of a competition for the fuel utilization between carbohydrate and fat, or between glucose and fatty acids, right? Why we don't use fatty acids in the fat condition and use glucose, or why we don't use glucose and utilize fatty acids in the fasting condition. So there is sort of competition. That means, so for the regulation, for example, in the fat condition, carbohydrate utilization will be stimulated or increased, but fat utilization will be suppressed. So we're going to explain later why fatty acids utilization will be suppressed in the fat condition, and carbohydrate utilization will be increased in the fat condition, and vice versa for the in the fasting condition. Glucose utilization will be decreased, will be inhibited, and fatty acid utilization will be stimulated. Yes?
Is there any way you could go back a slide for a second? I just graduated from there.
Oh, sure, yes. All right. So something related to the glucose or the amino acids metabolism that goes down, right, in the fat condition, in the starvation, and some fuels that are related to fat utilization will be goes up in the fasting condition, right? Okay, so this is the Randle cycles. So Randle cycles, glucose, or some people also call glucose fatty acid cycles, right? Mean in fat condition, body utilize glucose, and in fasting condition, body utilize more fatty acids. And this two nutrient fuel utilization has competition in different physiological condition, right? So let's see in the fat condition, right, the right side of the slide. So we have a lot of glucose, right, available from the diet. So glucose levels, blood glucose levels increase after meal, and that which will stimulate insulin release and suppress the glucagon secretion, right? So the insulin to glucagon ratio is increased in the fat condition. So remember what insulin does is to inhibit the lipolysis, right? So when insulin levels increase, then the mobilization of fatty acids or lipolysis process will be inhibited, right? So then you have less fatty acids being released from adipose tissue, mean that you have less fatty acids available for oxidation for energy. So this will lead to increased glucose utilization by many tissues, right? So this sort of a simple mechanism, why in the fat condition our body utilizes more glucose and less fatty acids. The simple reason is because of the hormone change. So then regulate the availability of the fatty acids, right? So you have less fatty acids available or released from, the adipose tissue. So that will lead you to utilize more glucose for the energy. And during the fasting, because we don't eat food, right? We don't have much glucose being absorbed. So the glucose level in the circulation that decreased, right? And this decreased levels of glucose will reduce insulin release and then will stimulate glucagon release, right? So insulin to glucagon ratio will be decreased, right? In the fasting condition, have a lower insulin to glucagon ratio. And a similar mechanism when insulin levels decrease, and it's also involved in the catecholamine levels, right? I did not list it here. Catecholamine levels should be increased, right, during the starvation. And catecholamine is a major driver for the lipolysis, right? So, and combined with the decreased insulin levels, this profile of hormones will lead to increase the lipolysis, increase the mobilization of fatty acids from adipose tissue, right? So you have more fatty acids being released from the adipose tissue, have more fatty acids, available for utilize as alternative energy fuels. So then this will causing the decreased glucose utilization, right? So this is just a simple mechanism and how our body utilize different fuel in different physiological conditions. And again, it's usually regulated by hormone levels. Any questions about this Reynolds cycles or glucose fatty acid cycles? Right? Okay. And it's just some more information about the glucose fatty acid cycles. So see the muscle and adipose tissue, right? And this middle is the circulation plasma. So adipose tissue is a major source of the fatty acids, right? Provide, during the starvation and release the fatty acids. And muscle is a major tissue that utilize fatty acids during the starvation. So they put these two tissues together and see how the utilization being switched during feeding and fasting condition, right? So in the fat condition, we have a lot of glucose in the circulation, so both muscle and adipose tissue be able to take up a lot of glucose, right, and utilize glucose as energy fuels when produce the glycolysis pathway, the pyruvate. And in the muscle, that glucose can be completely oxidized for energy, right? And this utilization, glucose utilization, will inhibit the lipolysis, right? Inhibits the release of fatty acids as in the fat condition. And in the fasting condition, for example, when fatty acids levels increase, primarily released from adipose tissue and picked up by muscle. So muscle utilization of fatty acids increase, which then inhibits glucose utilization in the muscle, right? So the simple description of the Randle cycle of fatty acids and glucose fatty acid cycle is that when our body utilizes more glucose, then fatty acid glucose being inhibited, right, in the fat condition. And in the fasting condition, when our body utilizes more fatty acids and will inhibit glucose utilization, right? Right? Questions? Okay. And here the next two slides, I just want to go to a little bit detail in the sort of molecular mechanism, why and how our body suppress the glucose, suppress the fatty acid utilization when we utilize glucose during the fat condition, or how our body suppress the glucose utilization when we utilize fatty acids during fasting condition, right? So, and this slide just shows us the fasting condition, right? So, and the inhibition of glucose utilization by fatty acid oxidation. So in the fasting condition, we utilize lots of fatty acids, right? And we try to inhibit glucose utilization. So how does this happen? So you see that during the fasting, we have a higher levels of these fatty acids in the circulation, which initially or primarily released from adipose tissue, right? Because of insulin to glucagon ratios decrease and catecholamine levels increase, right? And glucose fatty acids can be utilized in the cells, and here we talk about skeletal muscle, right? And fatty acids utilization, first of all, fatty acids being activated to fatty acyl-CoA, right, in the cytosol, and then need to be transported into the mitochondria, and by controlled, and this process is controlled by this enzyme, right, CPT1, on the membranes of mitochondria. So control the transport of fatty acyl-CoA from cytosol into mitochondria. And then fatty acyl-CoA goes to the beta oxidation process, right, and produce a acetyl-CoA, which is a end product of fatty acid oxidation, beta oxidation, right? And remember, and let's look at the glucose utilization pathway. So glucose can be picked up by GLU4, right, in the skeletal muscle, and transported glucose into the cells and be phosphorylated to glucose 6-phosphate and to pyruvate, right? And pyruvate enters the mitochondria, and the next step for pyruvate metabolism is pyruvate being decarboxylated to acetyl-CoA, right? And this reaction, remember the enzyme is pyruvate dehydrogenase, right? Remember how pyruvate dehydrogenase is regulated by its substrate. Remember? one of the important substrates that regulate pyruvate dehydrogenase activities, acetyl-CoA, right? Still remember, right? So if acetyl-CoA level is increased, then will inhibit pyruvate dehydrogenase activities, right? Again, the pyruvate dehydrogenase is a key enzyme for the glucose oxidation, right? This is a key step, right? For complete oxidize, pyruvate need to be converted acetyl-CoA, then enter TC cycle, right? So pyruvate dehydrogenase is a key enzyme for glucose oxidation. So if pyruvate dehydrogenase being inhibited, then pyruvate cannot be further oxidized, right, for ATP production. Right? Only stop at the glycolytic pathway, right, produce the limited ATPs. So this is the one step or one mechanism for why increased fatty acid oxidation will inhibit glucose utilization oxidation, right? So again, the increased acetyl-CoA levels and inhibits pyruvate dehydrogenase and inhibits glucose oxidation. This is one mechanism, right? And if you look further, so acetyl-CoA being generated from beta oxidation of fatty acids, remember the pathways can be converted to citrate, right, in the TC cycles, and then citrate can be released into the cytosol, and citrate is an important substrate that can also inhibit the two key enzymes in the glucose metabolism pathways, right? One is the hexokinase, right? The key enzyme for glycolysis, remember, the first key enzyme. And the key enzyme is phosphofructose kinase 1, right? So these two key enzymes in glycolysis pathways can also be inhibited by citrate, right? So try to remember these two key substrates, citrate and acetyl-CoA. These two substrate levels significantly increase when fatty acids oxidation is increased, right? And also try to remember the three key enzymes can be inhibited by these two key substrates. One is pyruvate dehydrogenase, right? It's inhibited by acetyl-CoA, and hexokinase or glucokinase, and phosphofructokinase 1. Right? These two key enzymes can be inhibited by increased citrate levels. Right? So this is sort of a mechanism for why fat oxidation increase can inhibit glucose oxidation. Right? Any questions on this? Right. Okay, now let's look at the fat condition. So in fat condition, we have lots of glucose available, right? So we utilize glucose and we suppress fatty acids utilization. So same thing, the pathways for glucose utilization. And so for glucose utilization, again, pyruvate, generate pyruvate, and pyruvate being converted to acetyl-CoA, citrate, and this is a key reaction. The ACC, remember, sorry, not ACC. This is the reaction that converted citrate back to the acetyl-CoA, right? And here's a key reaction, sorry. So this is a key reaction, remember ACC, right? It catalyzes the formation of malonyl-CoA from the acetyl-CoA, right? So when glucose utilization is increased, you have lots of citrate acetyl-CoA being generated in the cytosol, and you also have a lot of malonyl-CoA being formed in the cytosol. So the malonyl-CoA level, which is a key in the fat condition, right? Malonyl-CoA is a key substrate, right, that can regulate fatty acids oxidation. Remember, this is a key reaction, right? The key enzyme that controlled by malonyl-CoA. Malonyl-CoA can inhibit the CPT1 activities and reduce the transport of fatty acids from cytosol to mitochondria for beta oxidation, right? So this again is the mechanism for why glucose utilization increase can lead to decreased the fatty acids oxidation, right? Again, this is a key substrate. Malonyl-CoA levels increase when glucose oxidations increase, and which inhibits CPT1 and reduce the transport of fatty acids and fatty acid oxidation, right? Questions? Yes?
Why doesn't citrate downregulate glucose like it does in fast?
Because in the fat condition, this ACC activity is increased. Remember, ACC activity is regulated by insulin, right? So this reaction is very high, so then you have a lot of, the citrate being removed, right, by this reaction. So you wouldn't have much high levels of citrate in the fat condition because of this reaction. It's very active, right? And in the fasting condition, ACC activity is low. So that's why you have a lot of citrate accumulated, right? You have a lot of, increased citrate levels. Good question. Any other questions? All right. Okay, so how does the body know when to switch metabolic fuels? We already mentioned the hormones, right? That's involved, in the regulation, like insulin, glucagon, and catecholamines, and also the regulate enzyme that control the metabolic fuel metabolism. Remember, we just mentioned the key enzymes, right? The CPT1, pyruvate dehydrogenase, and glucokinase, ACC, all these enzymes that involved in the metabolic fuel metabolism, then they can be controlled or regulated by substrate or by hormones. So then you control the whole metabolic pathway activities, and also the transporters that control the flux of metabolic fuels into the cells. For example, glucose transporters, right? GLUT4 is also the important point, regulatory point. Now I just wanted to talk about a little bit more about this. I think that this we have covered this earlier, right? But just wanted to remind you again, in the fasting condition, how our body to do something to maintain the glucose levels, and the glucose homeostasis or maintenance of blood glucose levels involves several tissues, multiple tissues, including pancreas, liver, muscle, and adipose tissue, are the four most important metabolic tissues that are involved in the regulation of a glucose homeostasis in fat and fasting condition, right? Let's talk about the fat condition. So we have ingestion of a carbohydrate, right, leads to increased glucose level. And the first reactive tissues is the pancreas, right? The increased blood glucose levels can stimulate insulin release. So insulin levels is increased, and this sort of second tier response you still remember, right? The muscle and adipose tissue are the most important tissue that involve the second tier response to the high blood glucose level. So insulin stimulates glucose uptake through the group 4 into the skeletal muscle and adipose tissue, right? So when you can pick up a lot of glucose from circulation, then reduce the circulating blood glucose level. And of course, the liver is also important tissue that also pick up the glucose, right? Which through the group 2 is the glucose-dependent glucose transporters, right? And a lot of glucose being also picked up by the liver and for making glycogen, right? Stored in the liver. So all these working tissues work together to reduce the blood glucose level after meal, right? To clean the blood glucose level, blood glucose. So now to allow the blood glucose to maintain at the normal range, right? And then starvation. So starvation, in those opposite conditions that we have a lower blood glucose level, which leads to less insulin release and more glucagon release, right? And this will also reduce the glucose uptake by muscle and adipose tissue, and also reduce the glucose uptake by the liver. So you have less glucose being uptake, but more. But on the other hand, because of glucagon levels increase, that can stimulate liver to break down glycogen, release glucose into the circulation, right? And also to make glucose through gluconeogenesis and release glucose into circulation. So again, you have a less uptake by muscle adipose tissue and more release from the liver, right, due to increased glycogen breakdown and increased gluconeogenesis. So all this again all contributed to raising the blood glucose during the starvation, right? And the other physiological, the third physiological condition is not fat fasting, but during the exercise, right? So during the exercise, the most responsive hormones that is actually catecholamine. So exercise can increase catecholamine, but also reduce the insulin levels, right? Because of the lower glucose, you know. So what catecholamine does is to increase lipolysis, right? And also increase glycogen breakdown in the skeletal muscle and amino acids catabolism, right? And increase gluconeogenesis in the liver, right? So the main function or role of a catecholamine is to, first of all, increase lipolysis, to release alternative fatty acids, right? To allow body be able to utilize fatty acids as energy. And the second important function of the catecholamine is to increase glycogen breakdown in the skeletal muscle, and also increase the amino acids metabolism to provide energy. And in the liver, catecholamine also stimulates the gluconeogenesis, right? So, so during exercise, it's also have some issues, body need to deal with, is try to maintain blood glucose levels and also increase the availability of fatty acids because muscle need to utilize both carbohydrate and fatty acids for making energies, right? And this slide I'm not going to go through again, but just you guys maybe, I think it should be very easy to understand, in the fat fasting condition, how liver contributed to maintaining blood glucose levels. And again, started with the hormone levels, primarily insulin and the glucagon. So see how insulin to glucagon ratio change and how this changes hormone regulates the liver, glucose metabolism like glycogen breakdown, glycogen synthesis, and gluconeogenesis, and how these hormones to regulate the production of glucose or glucose release, production and release of glucose from the liver, right, and to maintain the body, the circulating blood glucose level, right. So liver is also a very important organ and to help maintain blood glucose levels, right, by the two major pathways, actually three major pathways: glycogen breakdown, glycogen synthesis, and gluconeogenesis, right? And it's just mentioned again, the opposite regulate role of the insulin and glucagon in energy metabolism. So insulin basically is the anabolic hormone, right? So try to stimulate, synthesize the complex molecules or the synthesis of stored energy fuels. For example, insulin could stimulate glycogen synthesis, stimulate triglyceride synthesis, right? Lipogenesis and triglyceride synthesis. And also increase the protein synthesis. And glucagon is basically the opposite, it's all the catabolic hormones and increase during fasting, stimulate lipolysis, stimulate gluconeogenesis, and also the so I think we have several, I don't know how many, 3 tables, right? Just to list, the transporters, hormones, and pathways, particular key enzyme. You can go through all these 3 tables and to try to review, I guess, right, in different pathways, how they contributed to glucose homeostasis or energy metabolism in different physiological state, fat fasting conditions. Yeah, you have a question, right?
You said there's 3 tables?
3 or 2? 2? Ah, sorry, 2. I don't know why I said 3. 2 tables, right? One for fat and the other one for fasting condition. Okay, we have about 5 minutes. Let me see how many slides I want to go. For diabetes, right? Let's see. Okay. So now I'm going to talk about the diabetes, so disease condition, pathologic condition. So diabetes, you probably all know we have 2 types, right? Type 1 and type 2. So type 1 diabetes, the main cause is the insulin deficiency. So means they have some problem with the beta cells or pancreas, and the patient, type 1 diabetic patients, not able to secrete insulin, right? So they basically lack insulin. So it's also called insulin-dependent diabetes. And treatment for type 1 diabetes is simple, but just the supplement insulin injection. It can cure the problems. And type 2 diabetes is different. It's considered a metabolic disease. Its main cause is the insulin resistance. Insulin resistance means that the patient, they are able to release insulin, and the insulin level even higher than the normal people, but the type 2 diabetic patient, the insulin action is decreased. So the insulin cannot efficiently does its function, right, in the type 2 diabetes. So the insulin is called insulin resistance. And insulin levels high is kind of a compensatory, mechanism, because the insulin action efficiency decrease, so they try to make more insulin to compensate, the decreased action. But in the early stage of type 2 diabetes, the insulin levels high. But in the very later stage of the type 2 diabetes, insulin, they also have some problem with the beta cell, called beta cell failure. So later stage of type 2 diabetic patient, they also cannot make enough insulin. So they also need insulin therapy. For mostly, the treatment for type 2 diabetes, so we usually mostly just encourage them to change diet, to change your health lifestyle, do more physical activities and a healthy diet, but of course sometimes they also need to give them the anti-diabetic drugs, and particularly for the later stage, if they have some insulin production problems, they also need insulin therapy for type 2 diabetic patient, right? Okay. Do you guys have any questions about it? Okay, so I guess I will just leave the rest of the slide about diabetes next time, right?